Welcome back to the Deep Dive, where we extract the core logic from the world's most authoritative texts. If you are preparing for your health science exams or, simply want to understand the foundational chemistry that keeps you alive, this deep dive is for you. Today, we are plunging into the engine room of life itself, mastering carbohydrate metabolism.
And this material, which we're guiding primarily through the framework of Harper's Illustrated Biochemistry, is just so essential. Our goal today is to really demystify the core processes that govern your energy supply. So how your body takes in fuel, how it executes these massive energy shifts between meals, and how it stores that fuel for later. We really want to move beyond just memorization and get into the why behind every single reaction.
Okay, so let's unpack this with some quick critical definitions first. When we talk about metabolism broadly, we're talking about, every single chemical reaction in the body. The source material lays out three fundamental categories of pathways to manage all this chemical chaos. What are they?
Right. We separate them based on their purpose and their energy requirements. You can sort of think of them as construction, demolition, and the main intersection. So first, we have anabolic pathways. Anabolism means construction. These are the pathways that synthesize large, complex molecules, think building proteins or stacking up massive glycogen stores. And because you're building something complex from simple parts, these pathways are always endothermic. They need a constant input of energy.
Building things costs energy. That makes perfect sense. So the reverse breaking things down, that must release energy.
And that's the second category, catabolic pathways. Catabolism means breakdown. These processes, which often involve oxidation, take large molecules and turn them into smaller ones, and in doing so, they release energy. They're exothermic. But that energy isn't just wasted as heat, it's captured. It's captured in the form of reducing equivalents like NADH and FADH2, and ultimately, as the cell's #1 currency, ATP.
And the third type connects these two sort of opposing forces.
That's the amphibolic pathway. Amphibolic means it serves both purposes. It's the true metabolic crossroads. It links the constructive, the anabolic, and the destructive, the catabolic routes. And the definitive example, which we'll get into in detail, is the citric acid cycle. It accepts fuel for breakdown, but it also provides essential building blocks for synthesis.
Okay, so here's the key question for our listener who might be heading into a clinical field. Why does this classification even matter? Why should a future doctor or nurse care if a pathway is exothermic or if a molecule is amphibolic?
Because understanding the normal rules, the energy balance, the inputs, the outputs, that's the only way you can recognize when the system is failing. Normal function involves these incredibly complex metabolic adaptations to just daily life. Switching from fed to fasting, meeting the crazy high demands of exercise, or navigating states like pregnancy. When metabolism goes wrong, whether it's from a simple enzyme deficiency or nutritional deficits or the profound hormonal chaos of diabetes mellitus, the signs of disease are really just deranged pathway flux. For instance, in severe diabetes, the body thinks it's starving even though there's glucose everywhere. And that triggers all the wrong pathways to fire up.
Okay, let's start with that big picture then, the fuel requirements.
Yeah.
Our sources tell us that a typical 70-kilogram adult needs a surprisingly high amount of energy every day, somewhere between 8 and 12 megajoules, which is, around 1,900 to 2,900 kilocalories. And our diet is really biased toward carbs and lipids.
It is, and that range is necessary because the energy demand is so variable. But the fundamental challenge is universal for everyone. Our cells need energy constantly, 247, even when we sleep. But we only eat in, discrete meals.
So the body has to be a master of resource management. It needs a massive organized storage system to bridge those gaps.
Exactly. And the body has 3 main reserve depots, each one optimized for different speeds and durations. For quick access, we store carbohydrates as glycogen, mostly in the liver and muscle. For massive long-term storage, we use triacylglycerol in adipose tissue. This is by far our largest fuel reserve. And then finally, the source mentions labile protein stores. And while we don't want to use protein for energy in a severe calorie deficit, the amino acids from continuous protein turnover become an unavoidable sort of last-ditch fuel source.
And that triage mechanism immediately shows the consequences of eating too much or too little.
Oh, the outcome of consistent imbalance is just biologically relentless. A consistent caloric surplus is stored overwhelmingly as triacylglycerol, and that leads to obesity. A consistent deficit, though, first depletes your fat and carbs, and then the body turns to its own protein stores. And those amino acids, which are desperately needed for essential protein synthesis, they get diverted for energy instead, and that leads to wasting, emaciation, and eventually organ failure.
Let's dive into the most critical metabolic shift then, moving from the fed state to the fasting state. If I've just had a big meal, what is the body's simple priority?
In the fed state, your blood is just rich with absorbed nutrients. Glucose is the ample fuel, and high insulin signals to pretty much all tissues, especially muscle and adipose, to just happily take it up and use it or store it. It's a time of abundance.
But the moment we transition to fasting, the logic just flips 180 degrees. The new priority is protection, right? Specifically protecting two crucial, highly dependent organs.
That's the key triage. In the fasting state, the central metabolic goal is to spare glucose. Glucose has to be reserved for the brain, the central nervous system, which is largely glucose dependent, and crucially for the red blood cells, or erythrocytes. Erythrocytes are wholly reliant on glucose because they lack mitochondria. So they can't oxidize fats, they can't use the citric acid cycle, all they can do is anaerobic glycolysis.
So how does the body enforce this glucose-sparing policy? It has to force every other tissue to switch its fuel source.
Precisely. The non-essential glucose users, so liver, muscle, most peripheral tissues, they rapidly switch their preferred fuel from glucose to fatty acids. Fatty acid oxidation then ramps up dramatically.
And what role does the liver play in supporting those other organs that still need a usable fuel besides glucose?
The liver becomes a factory for alternative fuel. When fatty acid oxidation is high, the liver synthesizes and exports ketone bodies, acetoacetate, and 3-hydroxybutyrate. These are water-soluble molecules that can be used effectively by muscle, kidney, and even the brain after a few days of fasting. So this effectively means that fatty acids, which can't be used by the brain, are converted into a usable energy source that spares the glucose still in the blood.
That sounds like a perfect system. But what happens when the easily accessible fuel tank, the glycogen, runs dry? The sources say this happens after about half a day of fasting.
That's the emergency phase. Once hepatic glycogen is nearly depleted, around 12 to 18 hours, the body enters full gluconeogenesis mode. That's the process of making new glucose from non-carbohydrate sources. The key substrates for this are amino acids, scavenged from protein turnover, and glycerol, which is released during lipolysis in adipose tissue. And this protein breakdown is really the most metabolically expensive part of that fasting shift.
It sounds like this entire complex regulatory ballet is controlled by just a handful of chemical messengers.
It is entirely governed by the interplay of hormones, primarily insulin and glucagon. They are the master conductors. Insulin signals fuel abundance, glucagon signals fuel scarcity. And this system requires both sufficient hormone production and the tissue's ability to respond to that hormone.
So when we talk about the clinical relevance, something like diabetes mellitus, we're really talking about a catastrophic failure of this hormonal triage system.
Absolutely. Impaired insulin production, type 1, or impaired tissue sensitivity, type 2, results in the body constantly getting starvation signals. The body just keeps mobilizing fat and protein reserves, leading to persistent high blood glucose because tissues can't absorb it, massive fat mobilization, and excessive production of ketone bodies, which can lead to ketoacidosis. The entire engine just breaks down.
If we zoom into the molecular level, there's a great unifying principle here. All the major breakdown products, glucose, fatty acids, amino acids, they all have the same ultimate destination.
They all flow to a single 2-carbon molecule, acetyl-CoA. Acetyl-CoA is the catabolic choke point. It's the final common pathway for oxidation. Once it's formed, this molecule enters the citric acid cycle for complete oxidation, yielding CO2 and generating the energy needed for ATP synthesis.
Since our focus is on carbohydrates, let's be specific about the path that glucose takes to get to that final common molecule.
The primary path is glycolysis, which converts the 6-carbon glucose into 2 molecules of the 3-carbon pyruvate. From there, the road forks based on oxygen. In aerobic tissues, pyruvate is oxidized to acetyl-CoA, ready for the citric acid cycle. In anaerobic conditions, like in red blood cells or muscle during a sprint, pyruvate is reduced to lactate.
But the importance of glucose isn't just about making acetyl-CoA to burn it. Glucose metabolites are fundamentally anabolic building blocks, too.
That is so often overlooked. Glucose metabolites fuel massive anabolic processes. They synthesize glycogen for storage. Through the pentose phosphate pathway, or PPP, they generate 2 absolutely essential components: NADPH, which is the reducing power needed for synthesizing fatty acids, and ribose, the crucial sugar for nucleic acids. And beyond that, intermediates provide the glycerol backbone needed to make triacylglycerols. Even some amino acids get their carbon skeletons from pyruvate. It's a true central material depot.
Okay, now we follow glucose down its main road, glycolysis. This is a foundational pathway, universal across all life, happening right there in the cytosol. And its ability to function both aerobically and anaerobically is, well, a metabolic marvel.
That dual function is absolutely key to cellular adaptability. You have tissues like hardworking white skeletal muscle fibers that need massive bursts of energy before the circulatory system can even supply enough oxygen. And then you have specialized cells like erythrocytes that are completely dependent on it because they injected their mitochondria during development.
Let's focus on that anaerobic side for a second. It sacrifices massive efficiency for pure speed and survival. What's the actual chemical benefit of making lactate?
Well, when oxygen is scarce, the mitochondrial respiratory chain stops running, and NADH can't be oxidized back to NAD plus do. But glycolysis requires a constant supply of NAD plus to keep processing glucose. So the desperate cellular fix is to use the accumulating NADH to reduce pyruvate to lactate, and that regenerates the vital NAD plus gus. It's a survival loop that keeps that minimal ATP flowing.
That makes lactate production not a waste product, but an essential part of the survival mechanism.
Exactly, but you pay that heavy price you mentioned, efficiency. A molecule of glucose yields about 32 ATP when it's fully oxidized aerobically, but anaerobic glycolysis gives you a net gain of only 2 ATP. And this low yield means the cell has to process glucose at a furious, unsustainable pace to meet energy demands, generating large amounts of lactate. The equation is basically glucose plus 2 ATP goes to 2 lactate plus 2 ATP.
Since most of the 10 steps of glycolysis are technically reversible, the whole pathway must be regulated by a few key irreversible steps. The source materials stress these three major checkpoints. Let's start at the very beginning with the phosphorylation of glucose, where we have two different versions of the enzyme, hexokinase and glucokinase.
This is a beautiful example of differential tissue specialization. Hexokinase, which is found in almost all tissues, is the high-priority enzyme. It has a very high affinity for glucose, a low kilometer value. This ensures that even when blood glucose is low, essential tissues like the brain can constantly and effectively pull glucose out of the blood and trap it inside as glucose 6-phosphate. Its activity is also tightly controlled by its own product.
So hexokinase is highly efficient and easily satisfied. Now, glucokinase, found mostly in the liver and pancreas, is the opposite. It's what's slow and greedy.
It has a much lower affinity for glucose, a much higher apparent commmm. In the liver, this high commmm means glucokinase only really becomes fully operational when glucose levels are very high, specifically right after a carb-rich meal. Its function in the liver is to act as a buffer. It clears that postnasal glucose spike from the portal blood for storage, normalizing peripheral blood glucose.
And its role in the pancreatic beta islet cells is really the perfect fusion of biochemistry and physiology. It's like a molecular tripwire.
It absolutely is the glucose sensor. Because of its high commmm, the activity of glucokinase in the beta cell directly reflects the concentration of blood glucose. When glucose is high, glucokinase ramps up, accelerating glycolysis, which dramatically increases ATP production. This rise in ATP then closes ATP-sensitive potassium channels, depolarizing the cell membrane, which opens calcium channels, and that calcium influx triggers the release of insulin. The glucose level is translated directly into a proportional hormone response.
Okay, moving down the path, we hit the second and arguably the most critical throttle point, phosphofructokinase 1 or PFK1.
PFK1 is the primary gatekeeper. It is subject to incredibly fine-tuned allosteric control, constantly assessing the cell's, financial health. The regulation by ATP is fascinating. Normal cellular ATP concentration is actually high enough to inhibit PFK1 by about 90%. It means the pathway is always waiting for the cell to signal that it needs more energy.
How does the cell definitively signal that energy is desperately needed, overriding that ATP break?
It uses 5-AMP. This is the metabolic crisis alarm. You have to remember the adenylyl kinase reaction. 2 ADP molecules can make 1 ATP and 1 AMP. So a small drop in ATP concentration, say 10%, can lead to a massive relative surge in AMP concentration, maybe 400%. That AMP surge instantly relieves the ATP inhibition on PFK1, slamming the gas pedal down on glycolysis.
And the final checkpoint is pyruvate kinase.
This is the third irreversible step, responsible for the second paired ATP generated through what's called substrate-level phosphorylation. The reaction is essentially unidirectional, mainly because the product, enol pyruvate, spontaneously isomerizes into pyruvate, making the thermodynamic barrier to go backward just too high. It's another point of tight control, being activated by fructose 1-valent-6-phosphate and inhibited by ATP.
So we have pyruvate. Assuming we're in a tissue with plenty of oxygen, that pyruvate has a ticket to the citric acid cycle. But first, it has to pass through another gatekeeper complex.
It enters the mitochondria and meets the enormous pyruvate dehydrogenase complex, or PDH. This is a crucial, high-stakes reaction. The oxidative decarboxylation of the 3-carbon pyruvate into the 2-carbon acetyl-CoA with the loss of CO2.
I want to pause here and verify a foundational rule, the physiological irreversibility of that PDH reaction. This rule dictates the body's entire approach to fasting.
You are exactly right to stress this. The PDH reaction is physiologically irreversible. Why is this so vital? Because that irreversibility means that the two carbons entering the process via acetyl-CoA cannot be used to make pyruvate and therefore cannot be used for net glucose synthesis. This is the central reason why the body has to rely on amino acids and glycerol, not fatty acids, to fuel gluconeogenesis during prolonged fasting.
We mentioned earlier that the PDH complex is huge and needs a lot of cofactors. Can we highlight which ones are most essential and why they're clinically important?
The PDH complex requires 5 cofactors, many from B vitamins: thiamine diphosphate, B1, lipoate, NAD plus furea, FAD, and CoA. If we focus on thiamine B1, it's essential for the decarboxylation step. A deficiency here critically impairs PDH function. And since the brain relies so heavily on glucose and the PDH gate is blocked, the result is a massive pyruvate buildup, which gets shunted to lactate. That causes severe neurological disturbances like Wernicke-Korsakoff syndrome, which you see in chronic alcoholics who often have severe thiamine deficiency.
So how does the cell regulate this massive irreversible commitment step?
PDH is regulated tightly by its products. High acetyl-CoA and high NADH shut it down, and also by covalent modification. It has its own dedicated kinase and a phosphatase. The kinase turns PDH off by phosphorylating it. The phosphatase turns it on by dephosphorylating it. And the kinase is activated when the cell is rich in energy-high ratios of ATP to ADP or NADH to NAD plus SANSO. If the cell has high energy reserves, it doesn't need to burn more carbohydrates, so PDH is inactivated.
The clinical consequences of glycolytic failure aren't just in neurological disorders. They're highly visible in the one tissue that is entirely reliant on this pathway.
The erythrocytes are the canary in the coal mine, for sure. Inherited deficiencies in essential glycolytic enzymes, particularly pyruvate kinase or aldolase A, cause hemolytic anemia. Since the red blood cell relies entirely on those 2 net ATP from glycolysis to maintain the ion pumps for its membrane integrity, losing pyruvate kinase causes the cell to swell and burst prematurely. This just illustrates how essential those final 2 ATP really are.
Okay, we've traced glucose from the mouth to the mitochondrion, yielding the crucial intermediate acetyl-CoA. Now we enter the core, the citric acid cycle, or TCA. We established its amphibolic. So, to verify my understanding, the cycle acts as both the ultimate furnace for every fuel type and the starting point for building new materials. Is that correct?
That is the essence of its pivotal role. It is the final common catabolic pathway, oxidizing all fuel inputs, carbs, fats, and proteins down to CO2 and water. But it is equally essential for anabolic routes, providing intermediates that serve as carbon skeletons for amino acids, starting points for gluconeogenesis, and necessary input for fatty acid synthesis. It truly sits at the crossroads.
Let's look at the mechanics. It's a cyclical process, so it involves an acceptor molecule that must be constantly regenerated.
Right. The cycle starts with the 2-carbon acetyl-CoA combining with the 4-carbon oxaloacetate to form the 6-carbon citrate. This is catalyzed by citrate synthase. Oxaloacetate is the catalytic engine. It has to be regenerated through the rest of the cycle to keep the process running.
And what exactly is the cycle doing to acetyl-CoA during this rotation?
It's performing a series of highly regulated dehydrogenations and decarboxylations. The ultimate goal is to strip away high-energy electrons, the reducing equivalents, and release the carbon atoms as CO2. In one full turn, the cycle produces 2 molecules of CO2, 3 molecules of NADH, and 1 molecule of FADH2.
I want to circle back to that carbon loss detail, as it's essential for understanding why you can't make glucose from fatty acids.
Yes, this is a common point of confusion. Although 2 carbons enter via acetyl-CoA, the 2 CO2 molecules released during that specific turn do not come from those newly added carbons. They come from the oxaloacetate backbone carbons that were incorporated in previous turns. The newly added carbons are retained until later iterations. This ensures that when 2 carbons enter, 2 are immediately lost, maintaining that 4-carbon balance needed to regenerate oxaloacetate.
And what's the total energetic payoff per cycle?
The great majority of the yield comes from reoxidizing those reducing equivalents. The 3 NADH molecules yield about 2.5 ATP each, and the 1 FADH2 yields about 1.5 ATP. So that's approximately 9 ATP via oxidative phosphorylation. We then add 1 more ATP, or GTP, generated directly by the enzyme succinate thiokinase, bringing the total energetic yield to about 10 ATP per cycle turn.
Let's identify the key regulatory points, starting again at the beginning with citrate synthase.
Citrate synthase catalyzes that initial condensation, which is highly exothermic. The enzyme is subject to something called channeling, meaning the product, citrate, is usually passed directly to the next enzyme, aconitase. However, the presence of high ATP or high fatty acetyl-CoA concentrations signals a high energy state and inhibits citrate synthase, which slows the overall input.
And aconitase itself has a well-known vulnerability to specific toxins.
It does. Aconitase is inhibited by fluorocitrate, which is a metabolite derived from the poison fluoroacetate. This inhibition causes a dramatic accumulation of citrate within the cell, severely disrupting the entire pathway.
We mentioned that the iketoglutarate dehydrogenase complex is structurally similar to PDH. Does it require the same vital B vitamin cofactors?
It requires the identical 5 cofactors: thiamine, lipoid, NAD plus FAD, and CoA. So deficiencies in these vitamins halt 2 crucial gateways into the TCA cycle. This complex is also inhibited by its products, succinyl-CoA and NADH, and it's highly sensitive to arsenite, which poisons lipoate, and high concentrations of ammonia.
Wait, let's explore that ammonia inhibition for a moment. That is clinically vital, particularly in liver failure.
It is. High ammonia levels, which are characteristic of advanced liver disease, can cause severe neurological symptoms. Mechanistically, ammonia consumes iketoglutarate to form glutamate and glutamine, effectively withdrawing a critical intermediate from the TCA cycle. When you lose iketoglutarate, the cycle stalls, energy production plummets, and the brain suffers profoundly, contributing directly to hepatic encephalopathy.
Let's talk about the unique substrate-level phosphorylation step catalyzed by succinate thiokinase. Why is the form of the energy product, GTP, so crucial?
Succinate thiokinase produces either ATP or GTP. Gluconeogenic tissues like the liver and kidney have an isoenzyme that favors GTP production. And the use of GTP here creates a direct regulatory link to the amphibolic function of the cycle. Specifically, the enzyme PPCK and gluconeogenesis is GTP dependent. So the GTP generated by the cycle directly fuels the withdrawal of oxaloacetate for glucose synthesis.
That structural logic is brilliant. Now, how is the overall flow of the entire cycle controlled? Is it mostly about those localized irreversible steps, or is there a bigger global regulator?
The primary controller is global and elegant. It's called respiratory control. The activity of the entire cycle depends on the concentration of oxidized cofactors, NAD+ and FAD. And the only way to regenerate these is through the mitochondrial respiratory chain, which is entirely dependent on the availability of ADP. If the cell has a high energy demand, so high ADP, the respiratory chain speeds up, regenerating NAD plus S, and the TCA cycle instantly accelerates to match.
So low ADP or high ADP acts as the ultimate signal to run the TCA cycle harder. Beyond that, what specific allosteric signals fine-tune the flux?
We see clear high energy, low energy switches. Isocitrate dehydrogenase is activated by ADP, signaling low energy, and it's inhibited by ATP and NADH, signaling high energy. This ensures that if the cell doesn't need the energy, it shuts down the cycle at this early point, allowing citrate to accumulate, which can then be exported for fatty acid synthesis.
Speaking of that amphibolic function, let's categorize those two roles. We need reactions that fill up the cycle when intermediates are withdrawn, the anaplerotic reactions.
Anaplerotic means to fill up, and these are essential. If you withdraw oxaloacetate for gluconeogenesis, you have to replace it, or the cycle stalls. The most important replenishment reaction is catalyzed by pyruvate carboxylase, which converts pyruvate directly to oxaloacetate.
And what's the perfect metabolic signal to activate that replenishing reaction?
Acetyl-CoA. During fasting, fatty acid oxidation is high, producing massive amounts of acetyl-CoA. If the TCA cycle is running slowly because intermediates like OAA have been withdrawn, the excess acetyl-CoA immediately activates pyruvate carboxylase. This ensures that when the body is busy oxidizing fats, it also creates the necessary OAA to sustain the cycle and, crucially, to maintain gluconeogenesis.
And conversely, we have the cataplerotic reactions drawing intermediates out. Where does the cycle export its carbon skeletons?
The two major destinations are gluconeogenesis and fatty acid synthesis. Since all TCA intermediates can be converted to oxaloacetate, they're all potentially glucogenic. For fatty acid synthesis, acetyl-CoA is needed in the cytosol, but it's made in the mitochondria. So when citrate accumulates due to high energy, it's transported out and cleaved by citrate lyase in the cytosol, generating the acetyl-CoA needed for lipid construction.
Okay, we now move to how the body manages glucose reserves, how we store it when it's abundant, and how we manufacture it when we're starving. Let's start with glycogen, the carbohydrate filing system.
Right. Glycogen management really highlights the functional specialization of tissues. Muscle glycogen is self-serving. It comprises roughly 75% of the body's total store, and it's used exclusively by the muscle itself during activity because muscle tissue lacks the final enzyme necessary to release free glucose into the blood.
So muscle holds its own energy hostage, so to speak. The liver, conversely, is the central bank, exporting glucose to everyone else.
Precisely. Liver glycogen acts as the crucial reserve used to maintain stable blood glucose during the initial phase of fasting. And it achieves this because it possesses the necessary enzyme, glucose 6-phosphatase, which hydrolyzes glucose 6-phosphate to free glucose, allowing it to be transported out to the brain and red blood cells.
Let's detail glycogenesis, the synthesis. Glucose has to be activated before it can be polymerized.
After the initial phosphorylation and isomerization to glucose 1-phosphate, the key step is activation. G1P reacts with UTP, uridine triphosphate, to form the active, high-energy nucleotide UDP glucose, or UDP-GLC. This activation is highly favorable because the pyrophosphate that gets released is rapidly cleaved, preventing the reverse reaction. Then glycogen synthase links these UDP-GLC units via A1,4 glycosidic bonds onto a starter primer called glycogenin.
And the branching enzyme creates that dense, rapidly accessible structure.
Yes, the branching enzyme is vital for accessibility. It transfers a minimum of 6 A1,4-linked glucose residues from a growing chain onto a neighboring residue via an A1,6 linkage. This creates a highly branched molecule which maximizes the number of non-reducing ends, allowing multiple enzymes to work simultaneously for both rapid synthesis and equally rapid breakdown.
Now, glycogenolysis, the breakdown, this is a distinct process, primarily driven by a phosphorylation enzyme.
The rate-limiting enzyme is glycogen phosphorylase. It breaks the N1,4 bonds using inorganic phosphate rather than water, producing glucose 1-phosphate. It works sequentially until it hits a branch point. The debranching enzyme then takes over, which has two activities: a transferase and a 1,1,4,6 glycosidase, which cleaves the final 1,2,6 bond and notably releases a single residue of free glucose.
The coordination between synthesis and breakdown is the centerpiece of this chapter's regulatory story, relying on reciprocal phosphorylation.
This coordination ensures that when you break down, you stop building, and vice versa. The hormonal signal, like glucagon or epinephrine, activates the second messenger, CMP. High CMP activates CAN-MP-dependent protein kinase, and this kinase performs two synchronous antagonistic actions through phosphorylation. It activates phosphorylase kinase, leading to activation of glycogen phosphorylase, which breaks down glycogen, and it simultaneously phosphorylates and inactivates glycogen synthase, which stops synthesis.
So the same phosphorylation cascade provides the gas pedal for breakdown and the brake for synthesis.
Okay.
But the muscle has an emergency override that doesn't require the full hormonal signal.
That's the calcium specificity in muscle tissue, which is just pure evolutionary efficiency. When a muscle contracts, calcium ions are released. This calcium binds directly to the calmodulin subunit of phosphorylase kinase. This binding partially activates the kinase, synchronizing glycogen breakdown with the mechanical activity of the muscle, ensuring fuel is supplied instantly, even before the slow-acting hormonal cascade is fully triggered.
When these systems fail, we see the glycogen storage diseases. These clinical examples are crucial for understanding the functions of the specific enzymes.
They are critical. Take type IgSD, von Guericke disease. This is a deficiency in glucose 6-phosphatase in the liver. Since the liver can't release free glucose, the patient experiences severe fasting hypoglycemia, and the liver swells massively with accumulated glycogen. The exit ramp is blocked. Conversely, in type 2 GSD, Pompe disease, the issue isn't the central metabolic pathway, but a defect in a lysosomal enzyme. This leads to glycogen accumulating in the lysosomes, causing muscle weakness and cardiorespiratory failure.
Let's pivot now to the ultimate contingency plan, making glucose from non-sugar sources. Gluconeogenesis.
Gluconeogenesis is non-negotiable for survival. It synthesizes glucose from non-carbohydrate precursors, glucogenic amino acids, lactate, glycerol, and propionate. This process is absolutely essential for fueling the brain and red blood cells during prolonged fasting. The primary site is the liver, but during extended starvation, the kidney becomes a significant contributor, supplying up to 40% of the body's new glucose.
Since glycolysis and gluconeogenesis are opposing pathways, the body must have separate enzymes to bypass the three major irreversible steps of glycolysis. Otherwise, they would just cancel each other out in a futile cycle.
That's the entire engineering logic of gluconeogenesis. The first bypass addresses the pyruvate kinase step, and it requires two separate enzymatic reactions spanning two cellular compartments.
Let's follow that first bypass from pyruvate in the mitochondria.
Pyruvate enters the mitochondrion and is converted to oxaloacetate by pyruvate carboxylase. This reaction requires ATP and the cofactor biotin, and this is where we see that exquisite regulatory logic again. Pyruvate carboxylase is strongly allosterically activated by high acetyl-CoA, the very molecule produced when the body burns fats during fasting. This ensures that if fatty acid fuel is abundant, the liver is prepared to drive gluconeogenesis.
And oxaloacetate, once it's formed, has to be converted to PE before it can leave the mitochondria.
Well, not always. OAA can't easily cross the mitochondrial membrane, so it's often converted to malate, which can cross, and then back to OAA in the cytosol. The OAA is then converted to PEP by PPCK, and as we highlighted in the TCA section, this step is GTP-dependent, linking gluconeogenesis activity directly to the GTP generated by the citric acid cycle.
The next two bypasses determine which organs can actually perform gluconeogenesis.
Exactly. The second bypass uses fructose 1,6-bisphosphatase to reverse the PFK1 step. The third bypass uses glucose 6-phosphatase to release free glucose. Both of these specific phosphatases are present in the liver and kidney, but are crucially absent from muscle, confirming the muscle's role as a selfish fuel user.
Let's review the sources of substrates used for this process, specifically the cycles that clear metabolic waste.
We rely on glucogenic amino acids and glycerol, but also two cycles involving mobile fuels. The Cori cycle, or lactic acid cycle, deals with lactate. Lactate produced by high-intensity muscle activity or by erythrocytes is shipped to the liver, converted back into glucose via gluconeogenesis, which is an energy-costly process for the liver, and then released back into the blood for reuse.
And the second is the glucose-alanine cycle, which is often used as a mechanism to transport nitrogen out of the muscle.
Precisely. Alanine, formed in muscle by transferring the amino group from other amino acids onto pyruvate, is exported to the liver. The liver then removes the amino group, forming pyruvate, which is immediately used for gluconeogenesis. It's a mechanism for both providing a glucogenic substrate and safely moving potentially toxic nitrogen to the liver for urea synthesis.
We absolutely must reinforce the concept we discussed earlier, the rule that fatty acids cannot be used for net glucose synthesis.
This cannot be overstated. Since the pyruvate dehydrogenase reaction is irreversible, acetyl-CoA derived from fatty acids cannot be turned into pyruvate. The carbons are lost as CO2 in the TCA cycle. The only minor exceptions are odd-chain fatty acids, which yield a small amount of propionate. And propionate can be metabolized to succinyl-CoA, which is a TCA intermediate and therefore is glucogenic. But this is a tiny contribution overall.
Let's discuss the single most important mechanism for reciprocal regulation, the master switch that controls whether the liver is in building mode or burning mode. Fructose 2,6-bisphosphate.
This is the high-yield insight for liver metabolism. Fructose 2,6-bisphosphate, or F2006BP, is the ultimate allosteric regulator. It is the most potent positive activator of PFK1, the glycolytic throttle, and simultaneously the most potent inhibitor of fructose 1,6-bisphosphatase, the gluconeogenic throttle. Its concentration dictates everything.
And how is the concentration of the master switch regulated?
By a single remarkable bifunctional enzyme that has both a kinase and a phosphatase activity. In the fed state, high insulin leads to the dephosphorylated form of the enzyme, which favors the kinase activity-making fructose 2,6-bisphosphate. This stimulates glycolysis. In the fasting state, glucagon activates the CAMP cascade, which activates a kinase that phosphorylates the bifunctional enzyme. The phosphorylated form favors the phosphatase activity breaking down fructose 2,5-bisphosphate. This removes the brake on gluconeogenesis and removes the gas pedal from glycolysis. The pathways are perfectly and antagonistically synchronized.
This coordinated control is designed to minimize waste, but the source materials do mention that some concurrent activity exists, the so-called substrate or feudal cycles. Why tolerate this apparent energy waste?
Well, this residual activity, where opposing reactions run slowly at the same time, allows for two things. First, it enables extremely rapid response and fine-tuning of metabolic flux. Second, and critically, it allows the cell to achieve a massive acceleration instantly when needed. For instance, in muscle, maintaining a slight feudal cycle allows the rate of glycolysis to increase by a factor of 1,000 almost instantaneously when contraction begins.
Okay, beyond the primary management of glucose as fuel and storage, we have pathways that handle specialized needs and other dietary sugars. Let's start with the pentose phosphate pathway, or PPP.
The PPP is an alternative cytosolic path branching off glucose 6-phosphate. Its central function is not energy production. We have to emphasize that no ATP is formed here. The two outputs are purely anabolic and protective, the generation of NADPH for reducing power, and the synthesis of ribose 5-phosphate, a building block.
NADPH is often confusing because it sounds so similar to NADH. Can you distinguish their roles sharply?
Sure, NADH is catabolic. It is oxidized in the mitochondria to produce massive amounts of ATP. NADPH is anabolic. It is the necessary reducing power for almost all reductive biosyntheses, building fatty acids, synthesizing steroids in the adrenal cortex, and constricting cholesterol.
Let's trace the pathway's two distinct phases.
The first is the irreversible oxidative phase. Starting with glucose 6-phosphate, two oxidative steps occur, generating two molecules of NADPH and producing ribulose 5-phosphate. The first enzyme, glucose 6-phosphate dehydrogenase, or G6PD, is the critical rate-limiting control step.
And then the PPP either produces building blocks or sends the sugars back to glycolysis.
That's the reversible non-oxidative phase. Ribulose 5-phosphate can be isomerized into ribose 5-phosphate, which is a required precursor for the sugar backbone of nucleotides. Or, if the cell needs more energy than building blocks, the sugar skeletons are rearranged via enzymes like transketolase, which needs thiamine, and transaldolase to form glycolytic intermediates like fructose 6-phosphate.
Which tissues run this pathway actively?
Any tissue heavily involved in reductive synthesis. The liver, for fatty acid synthesis, adipose tissue, the adrenal cortex for steroid synthesis, and the lactating mammary gland.
But the most important clinical function of the PPP is its essential life-saving role in antioxidant defense for red blood cells.
The erythrocyte relies on the PPP because it's the sole source of NADPH in that cell. NADPH is used by glutathione reductase to keep glutathione, GSH, in its reduced state. GSH is then used by glutathione peroxidase, a selenium-containing enzyme, to neutralize dangerous reactive oxygen species like hydrogen peroxide. This entire chain is what protects the erythrocyte membrane from oxidative damage.
So if you disrupt the G6PD enzyme, you disrupt the red blood cell's ability to protect itself.
And that is the foundation of G6PD deficiency, or FIVism, the most common inherited enzyme deficiency globally. Impaired NADPH production means the red blood cells are highly susceptible to oxidative stress, triggered by things like infections, certain anti-malarial drugs, or even fava beans. This results in rapid membrane damage and acute hemolytic anemia.
We also have to account for two other common dietary excesses, fructose and galactose. Starting with fructose, its entry into metabolism is uniquely problematic because it bypasses the regulatory choke point of glycolysis.
That's right. Fructose is metabolized primarily in the liver, initiated by fructokinase, which converts it to fructose 1-phosphate. The problem arises in the next step. Fructose 1-phosphate is cleaved by aldolase B into two products that immediately enter the lower half of glycolysis, completely bypassing the primary regulatory PFK1.
So fructose becomes an unregulated flood of fuel that the liver has to handle immediately. What are the key metabolic consequences of this flood?
That flood of intermediates, especially excess acetyl-CoA and glycerol 3-phosphate, is rapidly shunted into anabolic pathways, specifically fatty acid synthesis and the esterification of triacylglycerols. This leads to increased VLDL secretion, contributing to hypertriglyceridemia, a factor strongly implicated in atherosclerosis.
And the unexpected link between high fructose intake and gout.
The speed of fructokinase is so fast that it consumes ATP rapidly, transiently sequestering cellular inorganic phosphate. This phosphate depletion accelerates the degradation of purine nucleotides, leading to increased formation of uric acid, the cause of hyperuricemia and gout.
The clinical distinctions here are essential.
We distinguish between essential fructosuria, which is benign, a lack of fructokinase. The serious disorder is hereditary fructose intolerance, HFI, which is a deficiency in aldolase B. Fructose 1-phosphate accumulates uncontrollably, trapping massive amounts of inorganic phosphate, severely depleting cellular ATP and causing profound hypoglycemia and liver failure.
And finally, galactose, derived from dietary lactose.
Galactose is phosphorylated by galactokinase. The next step, catalyzed by galactose 1-phosphate uridyl transferase, exchanges galactose 1-phosphate with UDP-GLC to generate UDP-Gal and glucose 1-phosphate. UDP-Gal is essential for synthesizing milk lactose, glycolipids, and glycoproteins. And the body can also synthesize galactose from glucose because the final enzyme, UDP-GL4 iparase, is reversible.
And the major clinical deficiency associated with this pathway is galactosemia.
The classic form is a deficiency in the uridyl transferase. Similar to HFI, the accumulation of galactose 1-phosphate severely depletes phosphate, leading to systemic issues including liver damage and mental deterioration. A unique feature here is that unphosphorylated galactose is converted to galactitol in the lens of the eye. And galactitol is osmotically active, causing water influx, which leads directly to cataract formation.
This has been an incredibly detailed journey. What stands out to me is how every single component of this system, glycolysis, the TCA cycle, gluconeogenesis, the PPP, is not a separate process, but a single, integrated, and highly regulated machine designed to serve the hierarchy of cellular needs, all coordinated by the opposing signals of insulin and glucagon.
If we synthesize this deep dive into three high-yield insights, they are this. First, appreciate the foundational structure. Metabolism is a compartmentalized, integrated system relying on the tight energy control of anabolic, catabolic, and amphibolic pathways.
Second, remember the ultimate priority, glucose triage. The liver is the key regulatory and altruistic organ, supplying blood glucose, while muscle is the selfish user. That differential capacity is what allows the body to survive fasting.
And third, focus on the irreversible regulatory checkpoints. Hexokinase, PFK1, pyruvate kinase, and the PDH complex are where all metabolic decisions are enforced, using sophisticated feedback loops, covalent modifications, and allosteric switches like the fructose 2-pol-6 bisphosphate mechanism to dictate metabolic flux perfectly.
Okay, here's where it gets really interesting, the final provocative thought for you to carry forward. The entire complexity of this metabolic network, involving phosphorylation, allosteric control, and enzyme induction, is essentially a sophisticated triage system designed to achieve one objective-preserving neurological function. It is a biological testament to the ruthlessness of survival, prioritizing the 3 pounds of thinking tissue, the brain, over the entire mass of muscle protein, which is cannibalized for fuel during prolonged starvation. Consider the immense physiological burden that gluconeogenesis places on the liver and kidney, forced to continuously synthesize glucose, often at the high energy costs supplied by burning fat, just to keep the lights on in the central command center.
It's a system built on sacrifice for the greater good.
A perfect description. Thank you for joining us on this deep dive into the core engine room of life. We wish you the best of luck in your studies and understanding the mechanics of your own body.